Hello students, today we start the remaining part of the previous video, that is techniques of micropropagations. In last video I discussed about the advantages of micropropagations. So let's start techniques of micropropagations. So micropropagation is nothing but a technique in that from plant cells or organ we can get the large number of plantlets in controlled condition with supply of nutritions. So how this happening? So in this technique there is the first point, that is the selection of plants. So selection of plant material. So in the floriculture there is the selection of flowering plant. So when we start the micropropagation, first important technique, that is the selection of plant. So selection of healthy plant, healthy disease free plant is very important. So first you have to select the flowering plants. Then next point is the explant preparation. Explant, that means the part of plant which are used for the micropropagation techniques. हम जो भी प्लांट जो हम प्लांट सिलेक्ट कर रहे हैं उसमें से कोई भी सेल्स या उसका कोई भी पार्ट हम एज अ एक्सप्लांट माइक्रोप्रोपगेशन के लिए यूज़ कर सकते हैं. तो उसमें this explant is nothing but the actual flowering plant material used for the micropropagation. So in that case we have to cut this plant material in different shapes like rectangles, triangles or cylindrical shapes, and then these used in the micropropagation techniques. Then third important step in micropropagation technique, that is the sterilization. So sterilization, that means the material which will be sterilized, that's why the it will be free from microorganisms. So the surface sterilization explants by the 0.2% mercury chloride is carried out. Then heat sterilization of nutrient medium, then UV light, a laminar flow for inoculation chamber sterilization. So in the sterilization there is the two parts. One part is of explant sterilization, other part is regarding the nutrient medium or the instrument which have used in this technique, that will also be sterilized, and this all technique will be carried out in the UV light and laminar airflow. So all instrument or your material that will be properly sterilized. If it is contaminated, then the micropropagation will be not happening. The explants will be damaged due to the infections. So for plant material, the 0.2% mercury chloride is used for sterilization, and also by the heat sterilization, nutrient medium for nutrient medium heat sterilization technique is used, and all this all this procedure will be carried out under the UV light and laminar airflow. Then fourth step is the inoculation. Inoculation is nothing but the actual placing of sterilized explant on the sterilized nutrient medium under sterile condition is inoculation. Inoculation simply that means the transfer of explant on the nutrient medium in sterile condition. जो हम एक्सप्लांट सिलेक्ट कर रहे हैं उसका हम जो हमारा न्यूट्रिएंट मीडियम है nutrient medium या तो in the slant or the petri dish. On this we can transfer this explant on this nutrient medium in the sterile condition, that is the inoculation. Then next stage in the micropropagation, that is the incubation. So incubation is the growth of explant into plant through callus under controlled condition is incubation. The diffuse light, 20-degree temperature, sterile incubation chamber are required for the process. Incubation, that means the simply it means the growing of the explant जो हम न्यूट्रिएंट मीडियम पे जो हम प्लेस कर रहे हैं, देन दिस जो भी स्लैंट है, स्लैंट को हम इंक्यूबेशन चेम्बर में रखते. इंक्यूबेशन चेम्बर, दैट मीन्स द कंट्रोल्ड रूम. इन दैट रूम द light as well as temperature and sterile condition will be maintained. So in that room the diffuse light are present, then temperature will be maintained in 20 degrees centigrade, and it will be, all room will be sterilized. On that the incubation, or which is also called as the incubation chamber, उस रूम को जो कंट्रोल्ड रूम है उससे हम इंक्यूबेशन रूम्स बोलते हैं. So in that incubation room we can place this plant, so the explant will be grow as a callus. एक्सप्लांट का जो हम एक्सप्लांट और जो प्लांट सेल्स हम टेस्ट ट्यूब में रख रहे हैं, वहाँ उसकी ग्रोथ होकर वहाँ पे कैलस फॉर्मेशन होता है, दैट इज़ द इंक्यूबेशन. देन reculturing of callus. So reculturing of callus into 100 to 1000 of culture medium is carried out. 100 to 1000 of plants are generated. Actually, what is reculturing? Reculturing of callus, that means after the growing of callus, that callus will also cutted in a number of pieces. Then these pieces are transferred on the new culture medium. So from callus we can get, from single callus you can get 100 or 1000 pieces. So each pieces will be transferred on different or separate test tubes. That's why from one callus we can get 100 or 1000 number of pieces. So each pieces then will be growing as a new plant. हम जो pieces जो हम टेस्ट ट्यूब में डाल रहे हैं उसको हम फिर इंक्यूबेशन चैम्बर में रखेंगे, तो उससे उसकी ग्रोथ होके वहाँ पे हमें हर एक पीस का एक एक प्लांटलेट मिल जाता है. सो फ्रॉम वन कैलस यू कैन गेट हंड्रेड्स और थाउजेंड्स नंबर ऑफ प्लांटलेट्स. देन नेक्स्ट स्टेज इज द हार्डनिंग. हार्डनिंग, that means the generated plants burden from lab to greenhouse slowly and gradually. So in incubation chamber, after the sufficient growth of the shoot and roots, then these plantlets will be transferred on the soil. Then after transferring on the soil or pot, that is called the hardening, then the pot will be transferred on the greenhouse or that means hardening, that means the plants will be burdened from the lab to the greenhouse. जो हम लैब में टेस्ट ट्यूब में प्लांट्स है वो हम सॉइल पॉट में ट्रांसफ़र कर रहे हैं, फिर वो थोड़ी दिन हम लैब में रखेंगे, फिर स्लोली हम उसे सनलाइट में एक्सपोज करते जाएंगे और फिर उसे हम ग्रीन हाउस में स्लोली जो कंडीशंस हैं उससे हम ट्रांसफ़र, जो लाइट है वो लाइट बढ़ाते जाएंगे और उस प्लांट्स को हम ग्रीन हाउस में प्लेस करेंगे. दैट मीन्स द उससे क्या होगा कि जो प्लांट्स है वो धीरे धीरे हार्ड हो जाता है और जो सनलाइट है, वाटर है, वो खुद से वो उस कंडीशन में स्टेबल हो जाता है. इफ़ यदि हम डायरेक्टली जो लैब से प्लांट्स निकाल के यदि ग्रीन हाउस में रखते तो दी प्लांट्स विल बी डाई. दे कैन नॉट मैनेज दी on the greenhouse conditions. So the slowly transferring of this process from lab to the greenhouse is very important. Then last stage is the cultivation. So a plant produced through micropropagations are used for the commercial cultivation. So इससे जो after hardening of the plants in greenhouse, then you can these plants you can commercially cultivated in natural environmental conditions. So these are the steps which are including in the micropropagation techniques. Mostly in the flowering plants, as you seen in most of these ornamental plants in markets. These plants are developed in the lab or through the tissue cultures, and they can then they can be exposed on the commercial marketing. पहले वो आप वो जो प्लांट्स फ्लावरिंग प्लांट्स रहते हैं वो टिश्यू कल्चर द्वारा उसे डेवलप किया जाता है, फिर ग्रीन हाउस में रखते हैं, एंड देन आफ्टर हार्डनिंग होने के प्लांट्स वो हमें मार्केट में दिखाई देते. सो दिस इज़ वेरी यूजफुल बायोटेक्नोलॉजिकल टूल्स that will be helpful in commercial levels. So at last I repeat again the points of these techniques of the micropropagation. So in the micropropagations, the first important plant, that is the selection of plant material. So selection of healthy and disease free plant which is very important. Then from this plant we can select the explant. That is, explant is nothing but the actual material which is used for the micropropagation techniques. So in the you can use explant in different shapes like rectangle, triangles or cylindrical shapes. Then next important plant before going to actual inoculation, the sterilization is very important. Sterilization, that means the material will be free from the microorganisms. So for this you can use the surface sterilization, heat sterilization, etc. For explant you can use the surface sterilization by the mercury chloride in 0.2%, and the nutrient medium will be sterilized with the heat sterilization techniques, and also these all will be carried out under the UV and laminar airflow. Then inoculation stage. Inoculation is nothing but the actual transferring of the explant on the nutrient medium in sterile conditions. Then after inoculation, the incubation stage is open. In incubation, that means the transferred explant on the nutrient medium that will be placed in controlled conditions of the light and light as well as temperature. So under the diffused light and 20-degree temperatures and sterilized chamber which is known as the incubation chambers. So after the growing of callus or for the growth of the explant into the callus will be happening in the incubation. Then again next stage of the micropropagation is the reculturing of the callus. So after the developing of callus, we can again under the sterilized condition, this callus will be cutting into different pieces. From one callus we can get 100 or 1000 number of pieces. Then each piece will be transferred again new culture medium and new separate slants. So from one callus we can get 100 or 1000 pieces, and these pieces will be grown as a new plantlets. Then after developing of plantlets, this hardening technique is important. Hardening technique, that means the plants will be burdened और harden from lab to the greenhouse. जो हम plants lab में develop कर रहे हैं, after nutrient medium, after shoot or root development, then these plantlets will be transferred on the pots. These plots will be slowly exposed on the greenhouse. Then after properly hardening of the plants, these plants will be cultivated or they will be sales for the commercial cultivation. So in flowering plants, these mostly the micropropagation techniques is used. I hope all of you are understood about the actual micropropagation techniques which I have discussed today's lecture. So now I stop here.